Hello dear students. In the previous class we learnt about the topics outbreeding devices in plants, pollen pistil interaction and about artificial hybridization. Now the next topic is double fertilization. So after pollination what happens is the pollen grain by absorbing moisture from the stigmatic surface it ruptures to form the pollen tube. Okay, so after pollination the pollen grain produces the pollen tube. Clear? Now all the contents of the pollen grain, which includes two male gametes, are transferred to this pollen tube, and later on the pollen tube it reaches the ovary by passing through the style region. Understood? Now after reaching the ovary, the pollen tube enters into the ovule through the micropyle and discharges its two male gametes into the embryo sac. Clear? So once again I'll repeat. After pollination, the pollen grain produces the pollen tube and all the contents of the pollen grain are now transferred to this pollen tube, and now this pollen tube it reaches the ovary and finally it enters into the ovule through the micropyle. There it discharges its two male gametes into the embryo sac. Okay. Now of these two male gametes, one of the male gamete fuses with the female gamete and forms a single cell structure called zygote. Okay, what happens of these two male gametes which are found in the embryo sac? One of the male gamete fuses with the female gamete. This fertilization or syngamy takes place. As a result of fertilization, a zygote is formed and this zygote is diploid. Okay. Now the other male gamete it fuses with the secondary nucleus. Now how this secondary nucleus is formed? So the secondary nucleus is formed by the fusion of two polar nuclei. Clear? So the other male gamete it fuses with the secondary nucleus which is located at the center and forms the primary endosperm nucleus. Clear? And this primary endosperm nucleus is triploid in nature. Understood? Once again I'll repeat. Of the two male gametes, one of the male gamete fuses with the female gamete. Thus fertilization takes place. As a result of fertilization, zygote is formed. Okay. Now the another male gamete it fuses with the secondary nucleus. As a result of this, primary endosperm nucleus is formed, and this primary endosperm nucleus is triploid. Clear? As it involves the fusion of three haploid nuclei, it is called as triple fusion. Okay, and since two types of fusions means syngamy and triple fusion takes place in an embryo sac, the process is called as double fertilization. So why it is called as double fertilization? Because it involves two types of fusions. One is syngamy and the other process is triple fusion. Is that clear? Any doubts? Okay. And this double fertilization it is an event unique to the flowering plants. Clear? Okay. Now this is a picture of double fertilization where you can observe the deposition of pollen grain into the stigmatic surface, and now this pollen grain by absorbing moisture from the stigmatic surface it produces the pollen tube, and to this pollen tube all the contents of the pollen grain which includes two male gametes are transferred. Now this pollen tube it reaches the ovary and then into the ovule, and finally it discharges its two male gametes into the embryo sac. Okay, so in the embryo sac, here one of the male gamete it fuses with the female gamete, that means with the egg cell to form the zygote, whereas the other male gamete fuses with the secondary nucleus to form the primary endosperm nucleus. Okay, so here you can clearly observe the release of sperm cells or the male gametes. So by the fusion of male and female gametes, zygote is formed, and by the fusion of male gamete and secondary nucleus, endosperm is formed. Is that clear? And this process, that means the double fertilization process, it is an unique feature of the flowering plants. Okay, now coming to the next process, that is post fertilization events. So following double fertilization, what do you mean by post fertilization events? The events taking place after the fusion of gametes or after fertilization is called as post fertilization events. Following double fertilization, events of endosperm and embryo development, maturation of ovule into seed, and ovary into fruit are taking place, which are collectively termed as post fertilization events. Okay, so during post fertilization events, endosperm is formed, embryo is formed, and the ovule develops into seed, and ovary develops into fruit. So as I said before, the primary endosperm nucleus, which is formed by the fusion of male gamete and secondary nucleus, right? Now this primary endosperm nucleus, it develops into endosperm, whereas the zygote, how this zygote is formed? This zygote is formed by the fusion of male and female gamete. Okay, and this zygote develops into an embryo. So the primary endosperm nucleus develops into the endosperm, and zygote develops into an embryo. Okay, so during endosperm formation, what happens is the primary endosperm nucleus or the primary endosperm cell, it divides repeatedly and forms a triploid endosperm tissue. Once again, I'll repeat. During endosperm formation, the primary endosperm nucleus, it divides repeatedly and forms a triploid endosperm tissue. Okay. The cells of this tissue are filled with plenty of reserve food materials and are used for the nutrition of the developing embryo. So the function of the endosperm is to provide nutrition to the developing embryo. Is that clear? Now there are two stages in endosperm development. They are free nuclear endosperm and cellular endosperm. Now how this endosperm is formed? So when a male gamete, it fuses with the secondary nucleus, it forms the primary endosperm nucleus, which is triploid. Now this primary endosperm nucleus undergoes repeated division to form the endosperm tissue. Okay. Now about the free nuclear endosperm, here the primary endosperm nucleus, that means PEN, undergoes successive nuclear divisions to give rise to free nuclei. Okay, so during free nuclear endosperm formation, the primary endosperm nucleus undergoes repeated nuclear divisions to form the free nuclear endosperm. So here only the nuclear division takes place, and it does not contains the cytokinesis. That means cytokinesis does not takes place here. Okay, and example for this free nuclear endosperm, coconut water. Okay, liquid content or liquid endosperm. that is coconut water. Whereas the second type is cellular endosperm. Here subsequently cell wall formation occurs and the endosperm becomes cellular. And example for this, the white kernel in the coconut meat. Okay, so in the picture, this liquid content is the free nuclear endosperm, whereas the coconut meat or the white kernel in the coconut constitute the cellular endosperm. Once again, I'll repeat the free nuclear endosperm and cellular endosperm. Free nuclear means here the primary endosperm nucleus undergoes repeated divisions to form many nuclei, and here cell wall formation does not takes place. Whereas in cellular endosperm, immediately after the divisions of nuclei, cell wall formation takes place, so that the endosperm becomes a cellular here. Okay, understood? Okay. Now, after the fusion of male and female gametes, zygote is formed, and this zygote develops into the embryo. So, embryo is formed by the fusion of male and female gametes. After the fusion, zygote is formed and this zygote develops into the embryo. Okay. Now the embryo develops at the micropylar end where the zygote is situated. Now coming to the structure of the embryo, it is of two types. They are dicot embryo and monocot embryo. So let us move on to the structure of dicot embryo. The dicot embryo has an embryonal axis and two cotyledons. The portion of the embryonal axis above the level of the cotyledons is the epicotyle, which terminates with the plumule or the stem tip, and the cylindrical portion below the level of the cotyledons is the hypocotyle, that terminates with the radicle, nothing but the root tip, and this root tip is covered with a root cap. Okay, so once again I'll repeat the structure. So the dicot embryo, it mainly has two parts, the embryonal axis and two cotyledons. These two are the main parts of a dicot embryo, embryonal axis and two cotyledons. Now so these are the cotyledons. Okay, and this portion forms the embryonal axis. Now the portion of the embryonal axis above the level of the cotyledons, that means this portion constitute the epicotyle region. So it is the portion of the embryonal axis above the level of the cotyledons. Okay, and this plumule later on develops into stem portion, whereas the cylindrical portion below the level of the cotyledons, that means this portion constitute the hypocotyle, which terminates with the radicle, and this radicle later develops into the root, and the root is provided with the root cap. What is the function of the root cap? It protects the tip portion of the root system. Okay, so once again I'll repeat, the dicot embryo, it has got two parts, embryonal axis and two cotyledons. The portion of the embryonal axis above the level of the cotyledon, that means this portion constitute the plumule, which later develops into the stem tip, whereas the portion of the embryonal axis below the level of the cotyledon, that means the hypocotyle region, which later on forms the radicle, and radicle develops into the root system, and this root is covered with the root cap. So these are about the structure of dicot embryo. We'll move on to the next topic, that is structure of monocot embryo. The monocot embryo also possesses two parts, embryonal axis and single cotyledon, whereas in dicot embryo it consists of two cotyledons, right? And the cotyledon of the grass family, that means the cotyledon in monocot is called as scutellum. So what is scutellum? So scutellum is nothing but it is the cotyledon of the grass family, and this scutellum is situated lateral to the embryonal axis, and at its lower end the embryonal axis has the radicle and root cap enclosed with the coleorhiza, and the portion of the embryonal axis above the level of attachment of scutellum is the shoot apex enclosed in coleoptile. So this is the picture of monocot embryo. So this monocot embryo has got two parts, embryonal axis and a single cotyledon, and the cotyledon in this monocot is called as scutellum, and this scutellum is situated lateral to the embryonal axis. So this entire portion forms the embryonal axis. So the scutellum is located lateral to the embryonal axis, and the embryonal axis at its lower portion, it has the radicle and the root cap. Okay, radicle and root cap, which are situated below the embryonal axis. So this is the embryonal axis, and these two structures, means the radicle and the root cap, are protected by a layer called coleorhiza. Okay, whereas the portion of the embryonal axis above the level of attachment of scutellum is the shoot apex. So here is the shoot apex, and this shoot apex is protected by a layer called coleoptile. Okay, so these are about the structure of monocot embryo. Once again I'll repeat. The monocot embryo, it has got two parts, an embryonal axis and a single cotyledon. So here the cotyledon is called as scutellum, which is located on the lateral side of the embryonal axis, and the part of the embryonal axis above the level of attachment of scutellum is called as the shoot apex, which is protected by coleoptile, whereas the portion below the embryonal axis forms the radicle and root cap, which are protected by the coleorhiza. Am I clear? Okay. Now after fertilization, ovary develops into fruit. Okay, so this fruit is the matured or the fertilized ovary, and the wall of the ovary develops into the wall of the fruit, which is called as pericarp. So what do you mean by pericarp? So pericarp means it is the fruit wall or the ovary wall. So ovary wall later on develops into fruit wall, which is called as pericarp. Okay, and the fruits may be fleshy, for example guava, orange, mango, etc., or it may be dry like groundnut, mustard, etc. Okay, and the fruits are of two types. They are true fruit and false fruit. So once again I'll repeat. Fruit means it is the ripened ovary or it is a fertilized ovary, and the fruit wall, it is called as the pericarp, and the fruits may be fleshy or dry and are of two types, true fruits as well as false fruits. So true fruit means in this fruit develops only from the ovary. For example in grape, so grape is an example for true fruit. So So true fruit means if the fruit develops only from the ovary, okay, only from the ovary, then it is called as true fruit. Okay, whereas false fruit means here the fruit do not grow from the ovary. That means any other part of the flower other than the ovary develops into the fruit. So such fruits are called as false fruit. For example, apple, cashew, etc. So true fruit means it only develops from the ovary, whereas false fruit means it does not develop from the ovary but it develops from any other part except the ovary. Okay. Now in some species fruits develop without fertilization, and such fruits are called as parthenocarpic fruits. So what do you mean by parthenocarpic fruits? In some fruits like in banana, fruits develop without fertilization. Uh, that means this parthenocarpy can be induced through the application of growth hormones, and such fruits are also called as seedless fruits. Example, banana, watermelon, etc. So parthenocarpic fruits are nothing but these are the seedless fruits which are formed without fertilization. Okay, clear? Okay, now next, after fertilization, ovule develops into seed. So seed is the final product of sexual reproduction in angiosperm, and it is a fertilized ovule which are formed inside the fruit. A seed typically consists of seed coats, cotyledons, and the embryonal axis. Okay, so the cotyledons are the simple structure, generally thick and swollen due to the storage of food reserves. And based on the presence or the absence of endosperm tissue, the seeds are of two types: non-albuminous or non-endospermic seeds and albuminous or endospermic seeds. Okay, so what do you mean by non-albuminous or non-endospermic seeds? Seeds where the endosperm is completely consumed during the embryo development. Okay, so in some species of plants, the endosperm is completely utilized by the developing embryo, and in such seeds the endosperms are entirely absent at maturity. So such seeds are called as non-endospermic. For example, groundnut. Okay, whereas albuminous or endospermic means here seeds that retain a part of endosperm as it is not completely used up during the embryo development. So non-albuminous or non-endospermic seeds are nothing but these are the seeds which does not contains the endosperm tissue, whereas endospermic seeds means these seeds contain a small portion of the endosperm as it is not completely utilized by the developing embryo. Understood? And examples for albuminous seeds: wheat, maize, etc. Understood? And the integuments after fertilization get transferred into seed coats which provide protection to the young embryo. So these are the examples for endospermic and non-endospermic seeds. Groundnut is an example for a non-endospermic seed, and maize is an example for endospermic seed. So occasionally in some seeds the remnant of nucellus is persistent, and this is called as perisperm. So what do you mean by perisperm? So what do you mean by endospermic seed, and what is the function of endosperm? So this endosperm, it provides the nutrition to the developing embryo, right? So endospermic seed means some seeds possess small portion of endosperm at maturity. That means here the endosperm tissue is not completely utilized by the developing embryo, right? Likewise, in some plants, the nucellus, what do you mean by nucellus? It is also an nutritive tissue, right? So in some plant species, the nucellus is not completely utilized by the embryo. So, a portion of the nucellus is retained, and this remnant of nucellus is called as perisperm. So, perisperm is in the seed. In some species, in seeds, a small portion of the nucellus is retained, and this retained nucellus portion is called as perisperm. Okay, an example for this black pepper, beet, etc. So this is the image of a beet where you can clearly observe the endosperm tissue as well as the perisperm tissue. So perisperm is nothing but the remnant of nucellus or residual nucellus. So these are about the perisperm. The next topic is the seed dormancy. As the seed matures, its water content is reduced and seeds become relatively dry. So the general metabolic activity of the embryo slows down and the embryo may enter into a state of inactivity, which is called as dormancy. So dormancy means it is the inactive state of the seed or inactive state of the embryo. So when the seed becomes mature, what happens? The water content reduced so that the seeds become dry and the metabolic activity of the embryo slows down, so the embryo may enters into a state of inactivity. So this state is inactive state is called as dormancy. And in few species, the seeds lose viability within a few months, or seeds of many species live for several years. So what do you mean by viability period? So seed viability means it is the ability of the embryo to germinate. Okay, once again I'll repeat, seed viability means it is the ability of the embryo to germinate. So in few species, what happens? The seeds lose viability within few months, whereas in other species, the seeds may live for several years. For example, 2000 years old viable seed is of the date palm, which is discovered during the archaeological excavation at King Herod's palace near the Dead Sea. Okay, so these are about the concept of dormancy. Now the last concept is apomixis and polyembryony. So apomixis means it is the production of seeds without fertilization. So parthenocarpy means it is the production of fruits without fertilization, whereas if the seeds are formed without fertilization, then the process is called as apomixis, and it's a form of asexual reproduction that mimics the sexual reproduction. An example for this, some species of Asteraceae family and grasses, sunflower family. Okay, so apomixis means it is the production of seeds without fertilization, whereas polyembryony means in many species, for example in many citrus and mango varieties, some nucellar cells surrounding the embryo sac divide and forms the embryo sac, which produce the embryos, and thus each ovule contains many embryos. So this occurrence of more than one embryo in a seed is called as polyembryony. So what do you mean by apomixis? It is the process of production of seeds without fertilization, whereas polyembryony means it is the process of occurrence of more than one embryo in a seed. Okay, and examples for polyembryony, many citrus fruits. Okay, so dear students, by this we completed the second chapter. Thank you.